Oh, yes, this possibly could be technically live at the same time on YouTube. Again, it's just for a short period of time, as the full webinar will be for those who have registered. So, thank you very much for everyone who is joining me. And I will try and take you through this important subject that I've been trying to work on and come up with some clear explanations for. How to prepare for the next wave of COVID. For a lot of people, this may seem like a strange concept. Why would I still be talking about COVID when the pandemic is apparently over? Well, I think that based on what I've seen, and I can think based on what is happening across the world, the pandemic isn't actually over. And therefore, we should still be looking at strategies to be able to manage it. The reason I'm mentioning this to the general public is that I suspect that the strategy for most of the main agencies, health agencies across the world, has been primarily just to vaccinate. The problem is that my question has always been, what is plan B? If you can't get the results that you need through vaccination, what is it that the public can do? Is there anything? Or do they just have to accept that there is nothing left? And that's really all that I want to talk about. And I'll be explaining from a scientific perspective why I think this is important and why I think some people think some things work and what could be the mechanism of it. So let's start with some important points. The aim is not necessarily to prevent a SARS CoV 2 viral infection, but to reduce the risk of the virus getting into the bloodstream. This is a really important point. So many people are concerned, could you get an infection? That's not really the challenge. The challenge is how to reduce viral particles from getting into the bloodstream. That's really where the main point of what I'm focused on is around. So today I'm going to be going through the overview of the current state of COVID. I'll be looking at some upper airway strategies and critically pointing to why I think we need to be a little bit more flexible in our thinking. So, one of the areas, just for those who may get upset, I always talk about masks. But as usual, I will explain it from a scientific perspective. What you do with the information is completely up to you. We've heard about a lot of over the counter supplements as well as prescription medication. And I'll just be building out the science and explaining why some things work in the way they do. And then I'll share with you a few innovative ideas right at the end that I've been looking at that I think could be valuable as we move forward in the pandemic. So, here is a critical question that I think we need to reflect on. And this is just a touch on it, and I'll be coming back to it later on, and I'll explain why this is such an important concept. I believe that you have to have a number of layers of thinking. So, people usually just talk about mucosal immunity, but I'm speaking about effective mucosal immunity. What is it that prevents the virus from getting through this? Then, if there is a problem, oftentimes it can relate to interferon autoantibodies. And this was extremely important in terms of severe COVID 19. They found that up to 10% of cases had interferon autoantibodies. They are still very relevant in the context of recurring COVID infections. And then I talk about vaccination. There's a reason why I mentioned that in the context of protection and more risk of recurring infection. And it's largely because of the focused spike related response. I'm going to be doing a presentation at some point on one of the questions about what were the outcomes in countries that used whole virus vaccines. That's a really interesting question and one that I hope to look at in a little bit more detail. So it's not necessarily just about vaccination. The question is, do we have too much of a skewed immune response when we look at spike based vaccines? That's really the question that I'll be focused on as to why we could be having some of the issues. In terms of what I'd be focused on, strategies for targeting the virus and boosting defenses. Technically, I've broken it down into 7 categories: preventing viral binding, inhibiting viral entry, slowing endosomal traffic, inhibiting viral proteases, inhibiting the interferon blockage of the virus, inhibiting viral release, and just generally immune augmentation. And as I went through the list of things that I thought were important, I was trying to put each thing within the category that I thought it was relevant. Some things may have more than one strategy, and that's useful. But the essence of what I would say and what seems to have worked, and when I think about Honduras, which I'll be coming to later on, their success was about using multiple strategies to try and see if they could reduce the impact of the virus. I think that is extremely important. Going along, again, I will build out on this concept of the mucosal immunity. And this wall of immunity, that's the IgA antibodies in white, this wall of immunity that prevents the virus, even if it causes infection, to reduce the risk that it gets into the systemic circulation, that's into the lymph nodes and then into the bloodstream. And so that's another essential part that we are trying to see if we can explain. Then we have this point about mucosal immunity and the fact that it fluctuates. And this is really important to those people who are almost overconfident. You can find that people who are unvaccinated almost believe that they are invincible. You can hear that coming across. You just need to remember an important point here. Mucosal immunity will fluctuate based on your own characteristics. So you may find at particular times it's pretty good, quite strong, virus can't get through. But if you're stressed, if something else is going on, sometimes it could be down to medication, it could be all kinds of reasons. Your immunity drops, and that's when you then have a risk. I'm going to come back to this as to why this is so important. But the principle of mucosal immunity, I think, is exceptionally important and something that needs to be protected. So that's the end of the first section. And what I'd like to do is just make sure that I've covered any questions here that it could be valuable. And so Richard had asked, what if you have no tonsils or adenoids? Good question. I'm not sure, but I would presume that that would have an impact on mucosal immunity because these are critical parts of the mucosal protection in the upper airway. Just checking through some of the questions. How many evolutionary mutations jumps is BA 2.86 variant beyond the current vaccines? It's only just coming through. The work was done in Japan and China, and this is something that I think we will have to look at in some more detail in a little bit of time. Okay, good. I think that just look at one more point. How does a dry mouth caused by drugs affect mucosal immunity? I would think that's just down to mucus production. If you reduce mucus production, that's a critical part of the immune system being able to function. Okay, so let's go on to the next section. And in this point, I'll be talking about the current state of COVID. So, the current state of COVID is an important area for us to reflect on. And when we look at the information that is coming out at the moment across the world, I would think that it indicates that we're likely to have problems ahead. These are the current trends in the UK, and this is from the Zoe Health app, and this was just done in the last few days. And you can see that you had all of these peaks. This peak was in March of 2022. You then had another peak in July of 2022. And then in October, November, December, there were a few smaller peaks, and then it kind of waned down. And one would have expected by this point it would have waned right down and there would be nothing left. But what we're seeing here is an uptick in cases. And you have to remember, this is in an environment where measurements are not being done adequately. And so, within that perspective, this peak could actually be much higher than what we're seeing. So, in my view, that's an indication that we are likely to be in another problem. And if you remember, every summer when the virus seemed to dissipate or come down, everybody said the pandemic was over and then it would start again. I think we're probably having a similar pattern in the northern hemisphere. The southern hemisphere may be okay because they are moving into summer. So, this here are some of the infection trends from the USA. And at the moment, I don't know if you can see it quite clearly here, but you have the dominant variant is the XBB.1.5, and that's 53% of the cases. And you can see here in blue, this is from the nowcast estimates from the USA. And this was only up to about May of this year. So it's probably changing a little bit at this point. And I do have another one here. And so this one is more recent. And this one is showing that actually the main variant at the moment is EG5. This is one of the Omicron variants. And the one that seems to be growing fastest in the USA is the HV1. And this is up to the 30th of September 2023. So you can see there are lots of variants around. There's still a lot of virus spreading, and there seems to be a problem. I'm hoping that by the end of this presentation, you will have a better understanding as to how and why this mucosal immunity that I keep on referring to is such a critical piece of the puzzle. We have here, this was from the UK just a few days ago. Again, the new COVID variant being detected, which is the BA2.86. And the likelihood, because this variant they have found, this is the Chinese and the Japanese looking at it in vitro, so this is in the lab, it seems as though it evades most of the vaccine antibodies. And so this could be a problem. I mean, I say could be because the last time I thought there was going to be a big problem was last year with the BA2.75 was surging through Japan, and I thought that this was going to be a big issue. It didn't turn out to be, and so it's important to know that we can't actually be sure. But what seems to be occurring is that we're having more and more variants that are coming up that seem to have an impact with regards to the fact that the pandemic is not shifting. So, again, now to some basic sciences, the viral structure. And again, for anyone who doesn't remember this, this here is the virus, this gray ball on the surface in blue. Here are the spike proteins. This is the cross section of the virus, and this is the spike protein here. And then in orange here, you have these are envelope proteins, and in red, these are membrane proteins. Important to remember because, and you have the nucleocapsid protein here. So, usually, when you have a broad immune response from infection, all of these proteins are targeted, not just the spike. And that's one of the drawbacks with injectable vaccines is that when they're focused just on the spike, then you don't have as broad a response, and it's easier then for the variants to get around the immune system. So that has been one of the problems that I think the scientific community is cautious about acknowledging. I think that we just have to be quite honest about what is happening and reflecting on the importance of what is going on. So, moving along in terms of just breaking through the principles of an upper airway infection, this is again showing a head, cross section of a head. This is a nasal cavity here. And it's just showing these little dots here are virus particles that are going in. They should lodge somewhere up here where they'll replicate and then spread further down into the lungs or spread out to other people. This important image is taken from a paper, and it's called the SARS-CoV-2 replication in airway epithelium requiring motile cilia and microvilli reprogramming. This paper is huge when it comes to explaining how the virus does its infection. This is 6 hours. This is 24 hours. This is 48 hours. And we'll be coming back to this, but essentially, this is the virus entering. getting beyond the mucus, which is this cream area here. And so it gets to the tip, goes down the cilia, which normally sweep it, then they enter the cell. And then what happens is once inside the cell, some of the non-structural proteins impact on these tiny microvilli, which then become tree-like structures sticking out above the mucus and then spreading more virus, either outside or in within 48 hours. So it's a very, very efficient system that the virus has used, and it works very well, which is part of the reason why it's so hard to get control of the pandemic. Here we have an image, sorry, of a virus, and this is just giving you the principles based on that. Once it gets to the cell membrane, it will then get endocytosed, or there are different ways it can get in. But once it's inside the cell, it will then release its viral genome. That will then be translated into proteins, which will then become packaged, and then the virus comes back out on the other side. Thousands of viral particles. So one goes in, thousands come out at the other end. So again, a very efficient system of taking over full control of the whole protein replication in a cell. I'm now going to play a short video from Maastricht University, which I've kind of shortened, but it's a really wonderful video that I think will help to explain in a clear fashion what exactly happens with regards to how the virus actually works. So let's take a look at this, and then I'll come back and do some more explanation.
In our airways, the air we breathe comes into contact with a mixture of different types of healthy respiratory cells. Some respiratory cells have hair-like structures that move the mucus along. SARS-CoV-2 coronavirus targets these hairy cells. The virus itself is deceptively simple. It consists of only a few parts, but together, these parts create a highly effective virus. It has an outer membrane with 3 types of proteins on it: spike, envelope, and membrane proteins. Inside the virus, an assembly of nucleocapsid proteins hold together its genetic material, a single strand of viral RNA. The spike protein is responsible for making contact with the host cell and starting the infection. It has a coat of sugars that hides it from the body's immune system. Many of the variants of SARS-CoV-2 are characterized by mutations on this spike protein, seen here in red. These mutations can influence its function, some making the virus more infectious. Many mutations are unique to each variant, but some, like D416G, are found in each of the variants of concern. When the virus closes in on a host cell, its spike proteins open up and bind to a receptor on the host cell. The outer part of the spike protein is then cut off by a protein on the host cell membrane. This frees the inner core of the spike protein, which is cut open. The spike protein then unfurls and anchors into the host cell membrane, pulling the virus closer until the cell membrane and the viral membrane fuse, allowing the viral genome to enter the host cell. Inside the cell, a ribosome meets the viral RNA and starts to translate its genetic code. The result is a long protein chain containing non-structural proteins, or NSPs. Initially, these NSPs are all attached to one another, but some of the NSPs are able to cut the chain. The result of this cutting is the release of another NSP, which is able to grab onto a ribosome and occupy it in such a way that the ribosome can only read viral RNA. There, the membrane proteins are able to catch the nucleocapsid proteins and viral genome. This makes the membrane bend inwards, and a new viral particle eventually buds. when the membrane compartment containing the viral particle fuses with the outer cell membrane, the viral particles are then released from the cell. But that's only the first wave of SARS-CoV-2 particles that will be expelled. Eventually, the host cell produces so many viral particles that it dies and releases a whole wave of new virus particles into the surrounding tissue, ready to spread and infect nearby cells. This completes the life cycle of a single SARS-CoV-2 coronavirus.
That was an absolutely beautiful video, and I really credit Maastricht University for doing that. I think that that has explained a lot of what I know is very relevant as I go on to show you why certain things work in the context of the viral infection. At this point, sadly for those people who are on YouTube, this will only now be uh, they'll no longer be able to see the rest of the presentation. But for the others, I hope you can stay with me as we go through that breakdown of the viral infection and how it is relevant to which therapeutics can work in the context of COVID-19.